Hi, and welcome back to part 2 of our cell division section. We are going to start working on meiosis. We just finished mitosis, which is division of your somatic cells. You go from one diploid parental cell into two diploid daughter cells. So you start with sister chromatids, you go to the single chromatids, but it's still 46 chromosomes to 46 chromosomes. Here's where we're going to switch things up a little bit. Now we're getting into the gamete side of cell division. How do we make egg cells and sperm cells? Those are distinctly different cells in your body for a very good reason. We need that genetic variability in our population, or else if we're all the same, that's bad, because one bacteria or one virus can wipe out the whole entire population, and you don't want that. So the genetic variability that happens during these phases of meiosis are huge to allow us to survive and passing on of certain traits and all the wonderful things that you're going to learn about later in genetics. So let's talk about meiosis. This is going to be Roman numeral 4 in your notes, and we're going to do a little bit of just a little background, include a definition here and there for meiosis, and I'm going to teach this a little bit differently than I did mitosis, because meiosis resembles mitosis a lot. The same phases and stuff like that, but instead of going to 2 daughter cells, we're going to go to 4 daughter cells. So we're actually going to have 2 rounds of division in meiosis. So before we get any further in background, let's define what's actually happening here. So meiosis. Let's just kind of do another definition. We had one earlier, but let's refresh your memory. This is the production, and we're going to work backwards on it, the production of 4 non-identical daughter cells from one parental diploid cell. So the production of 4 non-identical, I'm going to say haploid gamete cells, get a little bit more beef to it, haploid gamete cells, and these are also daughter cells as well, from one parental diploid cell. All right. So in order for us to do this, to create 4 non-identical cells, haploid cells, we need to undergo 2 rounds of meiosis. And we affectionately call them Meiosis I and Meiosis II. And to show you how this all works, first I'm going to draw the cells out, and then we'll write things out instead. So if it's okay with you, I want to just show you what they look like before we put it into words as well. But before we do all this, let's talk and get a little bit more vocabulary under our belts. Let's do some background really quick. All right, so the parental cell that we're dealing with here. The parental cell is diploid, and you know this because we've talked about it many, many times. And because it's diploid, here's what it means. It has 2 copies of 23 chromosomes, for a total of 46, right? So that means part of me, because I'm diploid, all my somatic cells are diploid, I'm going to basically take all 46 chromosomes that I have and put a combination of them and package it up into 23 chromosomes to give off to my offspring. So here's what's happening. When we're made, we get, I'm going to use 2 different colors here so you guys can see what's going on here. When we're made, we get, oops, they're not like that. We get chromosome 1 from mom and chromosome 1 from dad. Pretend they look exactly the same. Chromosome 1 from mom and chromosome 1 from dad. That's actually 2 chromosomes because they're from different sources, but they're both labeled chromosome 1. You have 2 chromosome 2s, 2 chromosome 3s, 2 chromosome 4s, and so on for the total of 46 chromosomes that you have. Now, every time you see that same number, these guys are called homologous chromosomes. And here's why. Whenever you see that prefix homo in front, that means same. So homologous chromosomes, and here's why they're homologous chromosomes. The genetic makeup of the chromosome 1 I got from mom is identical to the genetic makeup of chromosome 1 from dad. Now here's what I mean by the genetic makeup. And I'm making this up, this part of it up. Let's pretend the genes that are encoded in your DNA, the genes on chromosome 1, let's say I have a hair color gene on here and I have an eye color gene. I'm making this up because I don't memorize chromosome genes. And I have a height gene that helps control how tall I am. If I have these genes on chromosome 1 from my mom, I have these identical genes on chromosome 1 from my dad. So I have hair, same location, eye, and height. And the thing is though, the genes are there. They're on the same location, they're doing the thing, but the coding for the gene might be different. And I know this for a fact because I've looked at my own stuff with my parents. My mom, this is my mom, so this will be mom and this will be dad. My mom gave me a brown color hair gene. My dad gave me a blonde hair color hair gene. And the reason you see brown is because brown is more dominant than the blonde, so that's why you see brown. But we'll get to that later in genetics. But what you need to know is that although I've inherited chromosome 1 from mom and chromosome 1 from dad, the genetic makeup of the chromosomes are identical. If we were to look at chromosome 2, you would see the same thing. The genes that are on chromosome 2 from mom are the same genes that are on chromosome 2 from dad. The coding of the genes might be different, but the information they code for, the end product, is the same. Either hair color, eye color, skin color, height, you name it, they're all there. And so all the qualities that make you you, they're all encoded within your DNA. You're more than just your DNA too. There's nature and other environmental factors that help code for, you know, all this stuff, how our personality works and stuff like that. It's a mixture of nature and nurture. But what you need to know is that our homologous chromosomes throughout the whole human population, our whole Homo sapiens, my chromosome 1 DNA, whatever spots of genes are on there, are also on your chromosome 1 DNA. That's what allows us to mate as Homo sapiens together. If we had different chromosome makeup, we wouldn't be able to mate, and we would make, there would be no fertile offspring coming off of this. It would just wouldn't work out well. So that's what a homologous chromosome is. Now, remember, when we're going through meiosis, the cell, same thing, just like before, had to go through G1, S, G2, and M. Excuse me. And so the chromosome is going to get copied in S phase, remember that? So during M phase, as soon as we enter meiosis this time, our homologous chromosomes are going to look just like they did for meiosis. So now, mom's chromosome 1 is made of sister chromatids, dad's chromosome 1 is made of sister chromatids. Again, we're starting with 46 chromosomes, but unlike in mitosis, these chromosomes didn't care about each other, they just did their thing. In meiosis, the homologous chromosomes are going to find each other. And this is where it gets beautiful and amazing and lovely and blows my mind. So just remember that this is this is chromosome 1, this is chromosome 1. You have that all the way to, 22, and then 23 are your sex chromosomes. So you have all these homologous chromosomes. You have a pair of each chromosome. That's why we are diploid organisms. The goal of this whole entire thing is to take this 43, chromosome 1 and chromosome 1, mix them up, essentially create a new chromosome 1 and hand that off to my offspring, right? That's why my kids, I have 2 kids, I have a daughter and a son, I see qualities from my parents in them because I am my parents. And so our traits keep getting passed down and down and down. So it's amazing. So that is kind of the background into how we're going to start meiosis. This word homologous chromosomes, I want you to be able to picture what we're talking about with homologous chromosomes. That's why it took some time getting into this. Okay, you ready to start meiosis? Let's go. I'm going to use 2 colors on this to signify homologous chromosomes because we're going to be going from 46 to 23 eventually, and you're going to see how these mix to create brand new chromosomes, like a new identity. So we're not making identical copies, we're doing non-identical cellular replication right now. So it's very cool. All right, hope you all got that. So here's going to be big B. This is going to be the phases of meiosis, and actually we're going to start with meiosis 1 because then we'll do meiosis 2. Meiosis 1 looks, you know, they all look like mitosis to a certain degree, but it's obviously very, very different. You're going to see why. So mitosis 1, and just like meiosis, we're going to use similar language, prophase, anaphase, metaphase. In meiosis, there's no prometaphase. Everything gets lumped into prophase. So let's start with meiosis 1. Let's first define what meiosis 1 is going to be. So as for meiosis 1, so here's what's happening. It's the separation of homologous chromosomes in a way. And so it's going to be the production of 2 haploid cells. We go from 2 to 4. That'll be meiosis 2. It's going to be the production of 2 haploid cells with non-identical sister chromatids. So we're going to work with this. Just go ahead and write this out. Production of 2 haploid cells with chromosomes made of non-identical sister chromatids. Okay. So what I would like to do here first is I'll draw the stages, and then we'll go back and write it all out. So this might be over a couple of videos, but you just got to bear with me. I promise it'll make sense. So I'm just going to erase this so I have a clean slate to work with. So number 1 under mitosis 1 is going to be prophase 1. So we have meiosis 1, and within each phase of within meiosis 1 you have your individual phases, and each one is preceded by the Roman numeral that you're referencing. So in this case, meiosis 1, we have prophase 1. After prophase 1, we have metaphase 1, and then anaphase 1, and then telophase 1. So you can see how this works. And if I say prophase 1, you automatically know we're talking about meiosis 1. When we get to meiosis 2, we'll have prophase 2. So it makes sense. If there's no Roman numeral, you know you're in mitosis, what we just learned about. So you don't have to worry about which one are we in. If there's a Roman numeral, you're in meiosis. If there's not, you're in mitosis. So hopefully that helps a little bit. So the first part, we're going to be in prophase 1. All right, so here's where we're going to need colors here because I'm going to draw some cells. And then actually, I may shrink that up a bit so I can draw a little bit more what's going on. Okay, I'm going to bring it over here. All right, so for prophase 1, very similar things that are happening in prophase 1 as did in prophase and prometaphase of mitosis. So same thing that happened in mitosis. In the end of G2, before we get into M, you have your centrosomes are duplicated. The nuclear envelope is breaking down. Microtubules are beginning to grow, do their thing, trying to break in. and they will in a second. But here's what's happening. Now, prophase 1 is amazing. Here's what's going on during prophase 1. The homologous chromosomes are finding each other. So this step takes a while to happen as well. And so what we're going to draw is 2 pairs of homologous chromosomes. Let's say we'll do chromosome 1 from mom and chromosome 1 from dad. So that's 2 chromosomes, right? And we'll do chromosome 2 from mom and chromosome 2 from dad. Remember, you were made of one from mom, 1 from dad, 2 from mom, 2 from dad. That's what you are. And so now we're finally allowing that DNA to mix up so you can go create a baby. That's exactly what's going on here. So for all of this, I'm going to put mom in black ink and dad in green ink. Okay? So chromosome 1 from mom is going to be right here, and chromosome 2 from mom will be right here. So this will be 1, and this will be 2. And then chromosome 1 from dad will be in green. But before I draw daddy, here's what's going to go on. During prophase 1, what happens is chromosome 1 from mom finds chromosome 2 from dad in the nucleus while the nuclear envelope is breaking down around it. And what happens is they do this amazing dance, if you will, called crossing over. They actually cross over, like the chromatids in the sister chromatids on each one cross over each other. And when they cross over each other, they're actually exchanging genetic material with each other. So here's what's neat. I'll draw it out. So here's chromosome 1 from mom, here's chromosome 1 from dad. And they're literally crossing over each other. I'm drawing it very simple, but what's actually happening is that if this is part of the arm from one of the chromosomes on dad, this is one of the chromosomes from mom, they're actually kind of intertwining together. And every place that they're able to cross over, the DNA coding is identical. That's why they're allowed to cross. The spaces where they don't cross, the DNA isn't identical. It's a little bit different, or it may be the same, you never know. But definitely the parts where they do cross over, the coding is the same there. So it's really, really neat. So what's happening here is that you get these homologous chromosomes, and they start to cross over each other, and we actually call this crossing over. And so you get crossing over of homologous chromosomes. And the region of where they cross over, the point where they literally cross over, we call that special region, move this up here, we call that a chiasmata. Chiasmata. That's the region of crossing over. And the other neat thing that we do, because you're seeing all these pairs being made. So if we have 46 chromosomes, what's going to happen is you're going to see 23 of these things being made. And what's happening, we call that when they cross over, when the homologous chromosomes cross over, this is 1 from dad, 2 from dad. When the homologous chromosomes cross over, we call this a tetrad. It's a tetrad because now there's 4 chromatids all hanging out together. Granted, the sister chromatids on mom's chromosome 1 are identical, the sister chromatids on dad's chromosome 1 are identical, but this is going to be the last time they're going to be identical. Because every time that DNA is crossing over, it's actually, they're exchanging genetic material with each other. And so we're going to see how this plays out when we get to metaphase 1, or actually we start getting to anaphase 1 and all that stuff. So I'm actually going to stop here because I don't want to impose on your time with the video, but I'm going to keep this on the board and we're going to keep moving forward. Don't worry, we're actually going to write stuff out, but first I want to draw it out and then we'll just write it out. Then we could write the story together. So this is prophase 1. Sure enough, we're going to start metaphase 1 when we get back. All right, I'll see you soon.